Our final speaker is Professor Kevin Gaston from the University of Nottingham, and he'll be talking about his work exploring protein kinase inhibitors and then related target identification. Kevin.
Thank you for the introduction. Thank you, everybody. I'll try and keep this brief because I know we all want coffee. And I feel a big fraud standing here because I know this is a big data session. I will touch very, very briefly on big data at the end, but mostly I'm going to be talking about small data. And that's our work on protein kinase ck2, which is my favorite kinase. But I will get to big data at the very end. First of all, I'd like to thank the AMMF for giving me the opportunity to tell you about this work and invite me here. And also thank my collaborators. Most of the work I'm going to talk about today was a result of a long-term collaboration with Jomnarong Lutsuan at the Chulalongkorn Research Institute in Thailand. That was a great collaboration that was funded by the Medical Research Council in the UK. And at the end, I'll talk about some work that we're now doing, which is funded by the AMMF. Jom and his work was ably supported by Eric van Kouwenberg in my lab, and also a great postdoc, Yudmar Marcolino, and some other people in my lab as well. And all of this is a joint project with Sheila Jayaraman as well at the University of Nottingham. So to start at the beginning, what is protein kinase ck2? ck2 is ubiquitously expressed. That means it's present in just about every tissue and every cell, and it's a protein kinase. It's an enzyme that adds phosphate groups to other proteins. And the interesting thing about this kinase is that it is upregulated in response to cell stress and also to growth signals as well. And it's important in a whole bunch of different things in the cell. I'm going to be talking mostly about DNA damage repair, so how DNA gets repaired in cells, and ck2 is very critical in this process. But it's also important in things like gene expression, immune signaling, drug resistance, and other things. So what's it got to do with CCA? Well, the point is that the activity of this kinase is increased in cholangiocarcinoma. So in cholangiocarcinoma cells, we have higher levels of ck2 messenger RNAs and proteins that are produced from those messenger RNAs. You can see that in this example here, where I'm showing some data from ck2 alpha, which is one of the subunits of ck2, and it has higher expression levels in cholangiocarcinoma, as it does in lots of different types of cancer. So ck2 is elevated in cholangiocarcinoma, and it's playing roles in those cells. And a few years ago, the FDA granted permission or orphan drug designation to a ck2 inhibitor, which I call CX4945 in this talk. But and this can be used in trials with patients with cholangiocarcinoma, and hopefully it will show some beneficial results, although that's not known yet. So what are kinases, just for the non-scientists in the audience? So this is an example of a protein here at the top. This cartoon shows a protein. Kinases are enzymes that take a phosphate from a molecule called ATP, and they transfer that phosphate onto proteins, and that can change the activity of those proteins. And those phosphate groups can be removed by a different class of enzymes called phosphatases. So we got interested in protein kinase ck2 because we study in our lab a protein called PRH, which is a transcription factor, and we discovered several years ago that PRH interacts with protein kinase ck2, which is the blue blob in this diagram. So protein kinase ck2 binds to our favorite transcription factor, and it phosphorylates the PRH protein. And that stops the PRH protein from being able to bind to DNA. The reason that's important in prostate cancer is because that results in increased cell proliferation. And the activity of ck2 is controlled by growth factors from the tumor environment. That signals through to ck2, it phosphorylates PRH, and then we get increased cell proliferation. So that's prostate cancer, not cholangio. But it highlights the point I'm trying to make here, which is that ck2 inhibitors are useful because they block the activity of ck2, and in this case, they block cell proliferation. But of course, we have to remember that they will also block DNA damage repair and lots of other processes that ck2 is involved in. So why is this important? Well, as you all know, the treatments for biliary tract cancer are gemcitabine and cisplatin. And cisplatin, for example, just to focus on that, is a DNA damaging agent. That's how it works. So in this diagram up here, we can see here's a piece of DNA, undamaged DNA. It reacts with cisplatin, and we get the formation of this DNA damaged product. And so that's how these chemicals work. So cisplatin, for example, induces DNA damage, and that results in cell death, and that's why it's useful in treatment. And if we take cisplatin and add it to cholangiocarcinoma cells, so in this graph here, you've got increasing amounts of cisplatin, and this is cell viability on this axis. So as you add increasing amounts of cisplatin, you start to reduce the number of viable cells, which is what you'd expect. It's a drug that kills cells after all. And the same thing happens if we use CX-4945, which is our inhibitor of ck2. As you add increasing amounts of the drug, it kills these cholangiocarcinoma cells. Two different cell lines here, one in red and one in blue. So both of these drugs will inhibit the viability of cholangiocarcinoma cells. So the question that John and I started to investigate was, will ck2 inhibition augment cisplatin activity? And John, who's very clever, decided to investigate this project in a sort of slightly complicated way, which I'll go through slowly. So what he decided to do was to either add both drugs together, so CX-4945 to inhibit ck2 and cisplatin, and then wait 24 hours and then see what happened. Or, more cleverly, because he's very clever, he decided to add CX-4945 first and then wait 6 hours and add cisplatin, or do it the other way around, which is to do the cisplatin treatment first, and then after 6 hours add CX-4945 and then see what happened. When we simultaneously add the drugs, it's quite boring. So what happens is, if we add CX-4945 here in this diagram, in this graph, sorry, up here is cell death. So we're measuring cell death, which is shown by these bars, two different types of cell death, but it's all cell death. We add CX-4945, we get an increase in cell death. That's not surprising. That's what I showed you on the previous diagram, really. If we add cisplatin, we get a cell death. And if we combine both drugs together, not surprisingly, we get an increase in cell death. So we get a combined effect of having both drugs. So it would be good to combine these drugs in treatment, we think, in patients. And hopefully the clinical trials will do this and show some added benefit to cisplatin and gemcitabine. But more interestingly, what John found was that depending on how you order these drugs, you get a different kind of response. So if we add CX-4945 first and then add cisplatin, we get a very, very small effect. We get very little combinatorial effect of these drugs. But if we add cisplatin first and then later on add the CX, then we see this very strong increase in cell death. So it's an interesting difference in depending on how you time giving the drugs. This is all in vitro, of course. And that happens in this cell line and also the same thing in a different unrelated cell line. So if we add cisplatin first and then CX, we see an elevated effect. And we can show that in a different way as well, because if we do a different kind of assay, this is a colony formation assay where we look to see the ability of cholangiocarcinoma cells to form colonies in vitro. You can see that with the untreated cells, which is called VC here for vector control, you can see we get lots of colonies. If we add CX-4945 followed by cisplatin, we get lots of colonies. But if we do it the other way around, cisplatin first and then CX-4945, we get a reduced number of colonies. And that's just quantitated in this graph over here. You can see that cisplatin followed by CX, which is in dark gray here, is more effective than the other way around. So it's an interesting point, I think. So we decided to look at this in terms of DNA damage. And we can measure DNA damage in lots of different ways, but the way that we chose to do it was by measuring the levels of a marker protein called gamma H2AX. Gamma H2AX is a protein which is produced in DNA damage factories in the cell where DNA is being repaired. And we stained these samples for gamma H2AX to look at DNA damage, and also with ck2 antibodies that bind to the ck2 protein. You won't be able to see it on these small pictures, but if you zoom in on the nuclei of these cells, you can see that there are these bright dots when we've treated with DNA damaging agents, and these dots are where the DNA damage repair is occurring in these DNA damage foci. So we can count the number of foci, the number of DNA repair factories in the cell, and we can also measure the levels of H2AX, just in terms of its fluorescence intensity. And if we do that, as you can see from this graph over here, where we've counted the number of foci, you can see that if we add cisplatin first, followed by CX4945, then we see this big increase in the number of DNA repair foci. And again, we can do, if we measure just gamma H2AX DNA repair protein levels, you can see the same thing. So that when we add cisplatin followed by CX, then we see this big increase in the DNA damage response. So just to summarize what's happening, so when we add cisplatin, we're getting DNA damage, as you would expect, and that can lead to cell death. But ck2 is getting in the way because it will initiate DNA repair, and that will reduce the efficacy of this DNA damaging agent. So the logic is that if we can inhibit the DNA repair pathway by inhibiting ck2, then we can augment cell death. So that's the story so far, very small data. What I'd like to do next is just to talk a little bit about, go into detail about how ck2 actually inhibitors actually cause cell death, and talk a little bit about the cell death mechanism. I hope you're with me so far. In other cell types, in my lab, we do a lot of work on prostate cancer. And in prostate cancer cells, things are very simple when we add CX4945, which is our inhibitor of ck2. In these kinds of graphs, we're looking at viable cells up here on this axis and drug concentration on the x-axis. And as we add the drug, increasing amounts of the drug, we see a reduction in the number of viable cells. So we can work out how good these drugs are at killing cells. And CX495 is very good at killing, these are human prostate cells and these are mouse prostate cells. It's a very good drug at killing cells. And the way it kills prostate cells is to induce a process called apoptosis. And we can measure apoptosis, which is a form of cell death, by measuring the amount of an enzyme called cleave caspase 3, because cleave caspase 3 is required for this apoptotic cell death. And you can see here that when we treat the cells with CX4945, we get a lot of big increase in the amount of cleave caspase 3. That's one of the assays that we use to say that this is apoptosis. You can also see down here that our favorite transcription factor, PRH, becomes less phosphorylated. So this antibody recognizes phosphorylated PRH, and you can see that there's a decrease in the amount of phosphorylated PRH in these cells, which is consistent with the stuff I was telling you about at the beginning, and an increase in the amount of unphosphorylated PRH. So in prostate cells, it's all really quite simple and really quite straightforward. Unfortunately, in cholangiocarcinoma cells, it wasn't so straightforward at all. What John did was that he looked at these cells to see how they were dying, or if they were dying. And what he found was that if we use this red dye here to stain dead cells, you can see, I hope you can see it, there's an increase in the number of red cells here that are the dead cells as we add CX4945. And he's quantitated that over here in this graph, so you can see that you get more dead cells as we add more of the drug, which is what we would expect. However, the interesting part was that when we then looked to see, well, is it the same as in prostate cells? Are these cells dying through apoptosis? It turned out that we could see no increase in cleave caspase 3. So the cleave caspase, here's the control over here in this dot over here. This is the control to show that the assay is working. But when we add the drug to these various different cholangiocarcinoma cell lines, we don't see any increase in cleave caspase 3. So that's really confusing. So you think, well, is the ck2 inhibitor really inhibiting ck2 in these cells? One way that we can measure that is to use an antibody that just recognizes all proteins that are phosphorylated by ck2. So these antibodies are not recognizing a single protein, they're recognizing all of the proteins in the cell that are phosphorylated by ck2. That's why you get a ladder of bands on Western blotting gels like this. And if we look at this ladder of bands, you can see that as we increase the drug, which is going across here, CX4945 being increased, we see a reduction in the phosphorylation of all of these ck2 targets. So ck2 is working in the cells, but it's not inducing cleave caspase 3 activity, and that's in multiple cell lines. And so John looked a little bit in more detail to see what was happening to these cells. And it turned out that the interesting thing about the cells was that they were starting to develop these very large white dots that you can see in these images. And these are called vacuoles. So most cells if they're just in culture or in the body, they don't have very large holes in them or very large vacuoles that you can sort of easily visualize. And that's shown in these pictures down here, where there's no drug. But as we add the ck2 inhibitor, 4945, you can see the formation of these very large, very visible holes or vacuoles. Vacuoles are fluid-filled spaces within the cell that are increasing, and you can measure that from these images either by looking at the number of vacuoles in each cell, and you can see here that as we increase the drug, we get a big increase in the number of these fluid-filled spaces in the cell, and also the vacuoles increase in size. So we're getting a big increase in these vacuoles. So what are the vacuoles? Well, it turns out that vacuoles are an indicative feature of a different kind of cell death, a kind of cell death called methiosis, which is when I was started off in this business, there was only two forms of cell death, and life was very simple. It was apoptosis and necrosis, and that's all you had to know about. But nowadays, there are at least 20 different kinds of cell death that occur in cells that we know about. Apoptosis is the one that I've studied for most of my career, but we also have this other process called methiosis, where the cells start to take in fluid, and then eventually the cells die because they've basically drunk themselves to death, and that's why it's called methiosis. Anyway, so these cells are not dying through apoptosis at all. They're dying through a completely different process called methiosis. So that made us wonder, well, when we're doing our cisplatin and ck2 inhibitor experiments, are we seeing methiosis? Well, the answer is, strangely, we're not. Because if we add cisplatin first, followed by CX4945, we don't see the formation of these vacuoles. So it's not really what was accounting for what we saw at the beginning of my talk. We don't see an increase in vacuoles, so we're not seeing methiosis in that condition. Strangely or interestingly, when we do it the other way around and we add CX4945 first and then add cisplatin, then we do see some methiosis, but not very much. So the methiosis is not really what's making these drugs work together very well in vitro. So that made us think, well, maybe this has got nothing to do with ck2 at all. Maybe this is something completely different that's going on. Maybe if we can get rid of ck2 in a completely different way, maybe we won't get this kind of cell death. So what we decided to do was to knock out the subunits of ck2 using a completely different approach, which is to use siRNA. siRNAs are small molecules that will take out messenger RNAs in the cell and prevent those proteins from being produced. So, for example, we can knock down ck2 alpha here, which is one of the subunits of ck2, using siRNA that reduces the protein expression, or ck2 alpha prime, because there are multiple different forms of ck2 in the cell. I haven't really gone into that. So we knock out ck2 alpha prime, and we also see a decrease in that protein. And we can then combine those different siRNAs to get a good knockout of the ck2 activity. And you can see that works here in this Western blot, where we're looking at multiple ck2 substrates, and you can see that the amount of these substrates goes down as we knock out the ck2 proteins. When we do that and we get rid of ck2 in this non-chemical way, or a way that's not using the inhibitor, then we don't see any methiosis in these cells. And if we change track completely and use a completely different ck2 inhibitor, this is a drug called TBB, which we often use in our lab to inhibit ck2 activity. So over here you can see that this drug inhibits ck2 because it removes these or reduces these ck2 substrates over here, these targets that are being phosphorylated. And so it's a very good inhibitor of ck2, but it doesn't induce methiosis. So life is complicated. So just to sort of summarize, because I hope I haven't lost you all, but I showed you this before. So we had our overall big scheme, which was to use ck2 inhibition to knock out ck2 activity, and that would enhance cell death. And that is what is being tried, not in our lab, but in clinical trials elsewhere. And those trials were intending to use CX4945 to do this. But the trouble is with this inhibitor is that it causes methiosis. It doesn't do what we thought it would do. And it's probably not to do with ck2. It's probably by a ck2 independent process, or it is by a ck2 independent process. And if we knock down CK2 activity in a different way using either TBB or other drugs or CK2 knockdown, then we can inhibit CK2 without causing methiosis, and that enhances cell death. So I just for the last 2 minutes, I'm going to change tack and talk about, get to the big data point that I was supposed to be talking about from the beginning. Sorry. Anyway, the tumor is a very complicated place, as Jesper has very ably demonstrated. It's got a lot of different types of cell types going on. And in our previous work on prostate, as I told you before, in my lab, we mostly study prostate cancer. But what we showed in prostate was that because tumors typically have very leaky blood vessels, they get a lot of things called blood platelets that are flooding into the tumor. And in prostate cancer, blood platelets signal through to protein kinase ck2 via a molecule called TGF-beta. And we published this kind of stuff quite a few years ago. Now, this is interesting because, of course, we were interested in using ck2 inhibitors to prevent cell proliferation. But if we know what these signaling molecules are that are coming from the microenvironment, then we can make better combinations of drugs. Because we can use, for example, in this case, in prostate cancer, we can use TGF-beta inhibitors in combination with ck2 inhibitors, and we can get more effective results. So what we're trying to do in collaboration, this is a collaboration with Dr. Isioma Abangwe, who's been mentioned already in this talk, and I should have mentioned her at the beginning, but in collaboration with Izzy at the University of Nottingham, what we're trying to do now is to identify all of the signals that control ck2 in cholangiocarcinoma cells. And we're taking the approach that Jesper mentioned earlier, which is an approach to use nanostring to look at all of the proteins and all of the mRNAs that are being expressed in cholangiocarcinoma. And then Izzy is segmenting the tumors to look at different regions to see what the signals are that control ck2. Unfortunately, and I'm very grateful to the AMMF for supporting this work, but at present the results aren't ready for prime time, so I'm going to hopefully show you those either next year or the year after. So just to summarize, we think, or I think that, or my team thinks that inhibition of ck2 is a promising strategy to increase the efficacy of cisplatin and other DNA damaging agents. And we think that using ck2 inhibitors is a really obvious and straightforward approach that would be very low cost and could be very effective. However, CX-4945, which is the lead candidate in this, has a complication, and the complication is that it induces methiosis, which could muck things up a bit in terms of how people think it works. But other ways of changing ck2 activity don't induce methiosis and are probably better for clinical trials. And finally, further work is needed to determine how ck2 is regulated in cholangiocarcinoma and therefore find better combinations of drugs that we can use to stop ck2. And as I said before, I'm very grateful to the AMMF for supporting that ongoing work. And finally, I'll come back to the end and just leave this here while I say thank you for your attention. Thank you.